get started here. So as you all know, MCI is a transitional phase when someone has diverged from the normal aging trajectory and is on the path to dementia. And the ability to accurately identify those individuals in that MCI phase is valuable from both a clinical perspective in terms of intervention and preventing decline, but also from a research perspective in terms of improving our prediction of who is most at risk and who's most appropriate to enroll in research trials and clinical trials. And so this is the line of research that I'll be talking about here, looking at MCI subtypes. So there are a number of different ways that MCI could be diagnosed. And these are the ways that I'll mention. So there's what we call the Peterson criteria or the conventional criteria that's widely used. And this is, looking at a subjective complaint from the participant or their informant, impaired memory, which is usually defined as impaired memory on one memory test, and intact activities of daily living. Then we have the consensus diagnosis, which is what's used at the ADRC. And that's where we have a group of experts that use all available clinical information. So it can be subjective assessment, objective assessment, neuropsych testing to arrive at a diagnosis. A potential limitation of these two diagnostic strategies is they do rely on subjective report as well as clinical judgment. So it could limit standardization kind of within and across, sites, clinics, research studies. And so this is one of the reasons that our group has really started to look at actuarial methods. So actuarial methods are those that are data-driven. They don't involve subjective information. We just really look at neuropsychological profiles. And so this can be done in a couple of different ways. One could be to use statistical methods like cluster analysis or latent class analysis to look at MCI subtypes, and that's what I'll focus on today. And another could be assigning an MCI diagnosis at an individual level. So many of you are probably familiar with what we call the Jack-Bondi criteria, where we're looking at an individual's scores and seeing if they have two impaired scores within a single domain. So kind of defining impairment at one standard deviation. And if they have this kind of consistent impairment within a domain, then that would be MCI based on that criteria. So different ways of identifying MCI. And so just to provide some background, I'm just going to spend about 10 minutes talking about some of the work that I've done in ADNI. Many of you may be familiar with this work, but it just kind of provides some context for setting up the ADRC study. So ADNI is the Alzheimer's Disease Neuroimaging Initiative, a longitudinal study around the US and Canada, and participants undergo neuropsych testing, CSF collection, and neuroimaging. So this is that conventional Peterson diagnostic criteria for MCI that I mentioned. And so you have your subjective complaint, objective memory impairment, normal general cognitive function, intact ADLs, and not demented. And the way that's operationalized by ADNI is through this. So in terms of criteria 2, 3, and 4, it's an abnormal memory function on one paragraph of logical memory, an MMSE score between 24 and 30, and a global CDR score of 0.5. So that's the method that ADNI is using to define their MCI subjects. And so what we did for this study, this was done in collaboration with Mark Bondi, Lisa Delano-Wood, and others. We pulled everyone out from that MCI cohort, so 825 individuals. And these individuals did get neuropsych testing, a brief battery. So we were able to pull out 2 measures of language, 2 measures of attention and executive function, and 2 measures of memory. And we put those into a cluster analysis. And so what's plotted here are Z-scores on each of those 6 measures. And what came out of the cluster analysis was 4 different subtypes. So the green bars are the language scores, the gray bars are the attention executive function, and the blue are the memory. So about 1/3 of the subjects were impaired on memory, had this isolated memory impairment. So we call them an amnestic MCI. 19% also had low memory, but they showed the significant impairment in the Boston naming task. So we labeled them the dysnomic MCI group. The dysexecutive mixed were impaired across the board on all 6 of those scores. And then a third of the subjects we called the cluster-derived normal group because they actually performed within normal limits on the neuropsych testing, despite their MCI diagnosis based on the Peterson criteria. So suggesting that these may be false positive errors. But importantly, all of these subjects were just classified as amnestic MCI by ADNI, but there do seem to be these meaningful subgroups within that larger group. And when we look at progression rates over time in those subgroups, kind of increasing means increased risk of progression to AD. So it was that dysexecutive mixed group that was most at risk, followed by the amnestic and dysnomic, and then the cluster-derived normal group at the bottom were much less likely to progress to dementia, and they actually were equally as likely to revert to a diagnosis of normal over the course of their time in ADNI. And then we've done a number of studies looking at different biomarkers and imaging profiles. So this is work done in Carrie McDonald's lab, where we looked at cortical thickness in the MCI subtypes relative to controls. And what's shown here is a comparison to normal. So the blue areas are areas where there was thinning relative to the normal group. So in amnestic MCI, we have this medial temporal lobe thinning primarily. In dysnomic, there's medial temporal as well as lateral temporal lobe thinning. Our dysexecutive mixed MCI, it has really widespread cortical thinning, which corresponds well to their kind of widespread cognitive deficits on testing. And then our cluster-derived normal group didn't show any cortical thinning relative to controls. And we've actually just recently published a follow-up to this where we looked at cortical thinning over 3 years. And we're able to see that false positive group really stayed healthy throughout the course of the 3 years. They didn't show cortical thinning over time. So the work from ADNI really kind of showed us that MCI can be empirically differentiated into these different cognitive subtypes, and that the subtypes are associated with different biomarker profiles and risk for progression to dementia. And then the empirically derived subtypes are also associated, as I mentioned, in a series of studies looking at CSF and neuroimaging, as well as risk of functional decline. Kelsey Thomas did a nice study looking at that. We've looked at PET imaging with Katie Bangan. And so a lot of work coming together really showing that breaking MCI down into different subtypes can be a useful endeavor in terms of figuring out different risk profiles. But one limitation of all of this is that our previous work was really limited by the very brief neuropsych battery, which was given in the ADNI study, and it doesn't really fully canvas all the cognitive domains that we could look at. So that brought us to the ADRC. And so what we did is apply these actuarial methods to the much more comprehensive data that's collected at the ADRC. And I actually have 2 projects going on right now that utilize ADRC resources. So the first is a career development award through the VA. We initially started off kind of looking at veterans primarily and have now expanded it to non-veterans. So we're able to include ADRC participants more broadly. And we're bringing people in and doing a comprehensive baseline assessment where we're collecting neuropsych, cerebral spinal fluid, neuroimaging, and then we've done some follow-up over the course of a couple of years. And I was just saying I was in the fortunate in terms of enrollment. We just finished recruitment in February before the pandemic hit, so it hasn't been too affected by that. So we're in the analysis phase of the CDA. And then I'm also working on through the ADRC REC, which is the research education component of the ADRC grant. This project looking more fully at the entire ADRC database and really trying to tease out these different subtypes within it. So that's what I'll focus on today. So the data I'm going to present is unpublished. We have a revision. The revision is now under review, so it's looking promising, but not quite published yet. So what we did is we examined all the participants in the ADRC dataset who were age 50 and older, either cognitively normal or MCI at baseline based on consensus diagnosis, and had sufficient baseline data that we could create some composite scores, which I'll show you in a moment. As you know, participants complete annual clinical, neurological, and neuropsych evaluations, and they receive a consensus diagnosis of either normal, MCI, or dementia. There's this other category called impaired, not MCI, which I'll mention a couple of times, which is applied to individuals where there is some impairment in cognition, but it's determined to be related to other factors in a participant's history and not necessarily neurodegenerative disease. So, it could be psychiatric condition or low baseline functioning, things like that. Okay, and these are the demographics of our sample. So we ended up with 738. They were about 72 years old, 14 and a half years of education, 54% female, and about 90% white. Okay, and this is what I'm going to go through today in terms of results. So first, I'll tell you about the cluster analysis and the subgroups that we found. We looked at rates of progression to dementia. We looked at types of dementia based on clinical diagnosis. In a subset of the sample, we had CSF biomarkers and neuropath data, and then we'll look at comparisons to the consensus diagnosis. So these are the measures that we included in the cluster analysis. So again, much more comprehensive than those 6 scores we had in ADNI, so that was nice. So broken up into seven different domains. And what we ended up doing was making a composite Z-score for each of these 5 domains. And as you can see, memory and learning and memory are overrepresented, just given the importance of that domain in early AD. And part of the composite scores was that we were able to include a lot more people, here and there would be missing scores, or, versions have changed over time at the ADRC. So being able to create those composite scores allowed us to include a lot more individuals. We did actually run it with the individual test scores in response to a reviewer comment and got a very similar profile, which was nice to see. And then we did a process of applying norms to all the neuropsych data using internal norms from the ADRC sample. So the raw scores on the neuropsych tests were converted to demographically corrected Z-scores. And this was based on performance of a robust cognitively normal group of 355 individuals. And by robust, what I mean by that is we identified those that did not progress to MCI or AD throughout the course of their participation at the ADRC. So we required that they have at least 1 year of follow-up, and at no point did they progress to any other diagnosis. And so using that group, then, and that group was quite well matched in terms of demographics to the overall sample. So very similar demographics to what I just showed. And so we made those composite Z-scores based on the performance of this group, and we corrected for age, education, and sex. Okay, so this is our cluster analysis. We did the cluster analysis and we got these 5 different groups. So what's plotted here are the mean Z-scores for each of the 5 cognitive domains. And I will walk you through the different groups here. So the first group we labeled optimal cognitively normal. So the dot in each of the violins there is the mean, with the line representing the standard deviation. And so as you can see on all 5 of the cognitive domains, this group is performing about a standard deviation or so above the mean. And that was 18% of the sample. The next was 28% of the sample, and their means are right around 0, so right around the mean level of performance. The next one is 14% of the sample, and we call this the non-amnestic MCI group. So that dotted line at -1 can help you see which ones are impaired there, but executive function in green and visual spatial in orange were the 2 that were low in this group. Then we had 29% with amnestic MCI, so the red memory bar is low, and then also they had low performance on language in purple. And then our mixed MCI group was 11% of the sample, and below 1 to 2 standard deviations or more on all of the 5 different domains. In terms of demographic differences, there were actually no age or sex differences between those 5 clusters. The mixed MCI group did have the lowest level of education at, it was 13 years versus 14 to 15. So still, a well-educated sample, but it was lower in the mixed MCI group. And then in terms of race, the MCI groups did have a higher percentage of non-white participants relative to the cognitively normal groups. Although, it is worth noting that it's largely a Caucasian sample, but there were some differences in there that would be worth exploring more. And then one factor that was a bit surprising is that there was actually no group differences in ApoE4 positivity for the sample. So we had ApoE for about 80% of the sample, but didn't see a difference between groups. And one potential reason for this, in our thinking about this, is that there may be a relatively high percentage of ApoE4 positive individuals in cognitively normal participants within the ADRC in general. So if you kind of think about who's more likely to volunteer for research, it might be someone that has a family history of Alzheimer's or potentially knows their E4 status. And so it may just be that it's overrepresented in the ADRC. And so we did look, if we look at consensus diagnosed normal participants, there were 31% that had E4 positivity. And then 34% if you look at our normal participants. But either way, it's more than in the literature. It's about 24% in European Americans. So that may be one reason for this different, lack of difference between the groups. So of the 738 participants, we had 23%, 172, who progressed to a diagnosis of dementia over time. And the dementia diagnosis was made on an average of about 6 years post-baseline, but with a very wide range between 2 years and 30 years. In terms of the amount of follow-up, the MCI groups had a shorter length of follow-up than the cognitively normal groups. So they were meeting criteria for dementia, sooner, whereas the cognitively normal groups had this much longer period of follow-up before they progressed. And you can see the overall progression rates here broken down by group. And there definitely were significant differences between these. The optimal and the typical cognitively normal groups did not differ significantly, but they were different from the non-amnestic MCI. And then we get kind of a stepwise increasing rates of progression as you go up. And I'll show you what that looks like in terms of a survival curve. So this is the survival curve showing risk of progression to dementia over time in our 5 cluster-derived groups. So as the groups go down, it means more individuals are progressing to dementia. So we have the optimally normal in blue, typical normal in green, the non-amnestic MCI is in red, right there in the middle, and then orange is amnestic and purple is mixed. And you can see down below the number of individuals that were included in the analysis at each time point. So there's quite a few individuals up until 10 years, 15 years, still a good number. It really drops off after that. And we could have arbitrarily cut it off, but we decided to just present what we had. But it is worth noting that those later time points have very few individuals. And so when we look at a difference in survival curves between the groups, there definitely are some significant differences. The one thing that's not significantly different is the 2 normal groups. Those are equivalent statistically, but all the others differ from each other. So the non-amnestic MCI participants, they're definitely not progressing at the same rate as the amnestic and mixed, but they are an at-risk group that we were able to pull out using this method. And then our mixed group is the most at risk. And the one thing about that non-amnestic group is, these are individuals who they're at risk, they may benefit from some intervention, but also they may get clumped into the normals. Typically they would kind of, contribute to some noise in a normal sample if we included those non-amnestic individuals. And then we did a Cox regression, just looking at risk of progression over time. And you can see the hazard ratios here are significant. And this was compared to the typical cognitively normal group. So about a 2 times risk of progression in the non-amnestic, like 3.5 in the amnestic, and 5.8 in the mixed MCI. Okay, so with regard to the type of dementia, so of the 172 who progressed, 84% of them, so most, received a consensus diagnosis of probable AD. So it is worth keeping in mind that was by far the most common diagnosis. But there were some participants that progressed to a non-AD dementia, or a mixed AD type of dementia. And the main point I want to make here is that the non-amnestic group was the one that was most likely to have individuals progressing to a non-AD dementia. So 30% of that group progressed to something else. You know, that it's only 7 people, but there were 2 that were vascular, 3 that had Lewy bodies, 1 with Lewy body and FTD, and 1 that was just indicated other CNS disorder. So more variable in terms of the etiology. But as I said, the majority of them were probable AD. But kind of interesting that, you know, that's the group that came out, and they're showing these more diverse types of diagnoses. Okay, and then in terms of cerebral spinal fluid, so cerebral spinal fluid was available for 27% of the sample. And this was 197 individuals. And lumbar punctures were conducted within 6 months of baseline testing for the vast majority of participants. So for 82% of the participants, they had their lumbar puncture within 6 months of the neuropsych testing, which is very impressive. I thought that was great. We did include individuals that had it up to a couple of years, just given literature out there showing that those biomarkers do not change very rapidly. They stay pretty stable. But there was no difference in between groups in terms of the average baseline to LP interval. In fact, it was 3 months on average. And what we were looking at is CSF markers consistent with AD, so beta amyloid, total tau, and then the ratio of tau over beta amyloid. And that's that ratio that's shown to be the most sensitive to differentiating, you know, clinical groups. And the cut point for that ratio was actually derived within the ADRC sample, a separate study where they looked at 462 participants that ranged widely from cognitively normal to severely demented. And that's how they were able to kind of figure out that cut point for where we should call the ratio positive or negative. And the cut point is very consistent with literature that's used kind of PET validation as well. Okay, so this graph shows the concentration of cerebral spinal fluid, beta amyloid in blue, tau in green, and then that ratio in red. And so what you can see for the blue bars, there's actually no significant group differences between those. I plugged the ends in here at the bottom. So there's 39 in the optimal group, 68 in typical, 36 in non-amnestic, 42 in amnestic, and only 12 in that mixed group. So we do have a very small number in the mixed group. So even though the mixed group is showing lower amyloid levels in CSF, which is a bad thing, you don't want it to be low, that difference was not statistically significant over the groups. However, for tau, that mixed MCI group, now the elevated level is bad. So they were showing higher levels of tau relative to the other groups. And the amnestic MCI group also showed a trend toward a higher level relative to optimal, cognitively normal. And finally, this is our ratio. And the dotted line shows that cutoff that was derived from the ADRC sample. And you can see that the mixed MCI group had the highest ratio compared to the first 3, so the normal groups and non-amnestic. And the amnestic MCI group had a higher level compared to optimal normal and non-amnestic MCI. So kind of some patterns in the direction you might expect, where amnestic showing some elevations in the ratio, and then that mixed group is showing an even higher level of abnormality. And then when we apply that cutoff and just characterize the groups as positive or negative, it was, again, that mixed MCI group that had the highest rate of biomarker positivity. So a small number of subjects, but definitely, you know, a very high risk group. So moving on to neuropath. So we had neuropathological data for 21% of the sample. The mean age at death was 86 years. And there's this very wide interval between baseline neuropsych testing and autopsy, and that did differ across groups. So for the two cognitively normal groups, it was about 15 years from baseline to autopsy. And then for the MCI groups, it ranged from about 7 to 12, with no difference in between them. So AD pathology was operationalized using consensus criteria, the NIA-Reagan consensus criteria for a postmortem diagnosis of AD. And what we ended up with were classifications of low, intermediate, or high. And that's referring to the likelihood that the cognitive impairment was due to the observed AD neuropathology. And so we also looked at then other pathologies like Lewy body vascular and other, which I'll mention. And a big thanks to Dennis Smirnoff for helping with all this. Dennis was very helpful in putting together the CSF and the autopsy data. And what's shown here is those three different groupings of high, intermediate, or low, kind of looking at severity of AD pathology based on that consensus criteria. So just kind of focusing in on the red, so that's the high severity. So the 5 cluster groups differed in the proportion of participants that were in the red here that had a high level of AD pathology. So specifically, the mixed MCI group showed the highest proportion of individuals with a high level, and that was relative to optimal cognitively normal and non-amnestic MCI. And then the amnestic MCI group had a higher level of high individuals relative to optimal cognitively normal. And the typical cognitively normal actually is a little more than both optimal and non-amnestic MCI. This typical group is kind of interesting. I think it could include some people that may have some subtle cognitive changes, along with some people that are normal. So I think it's interesting to kind of break these normal groups apart. What's important though about all those comparisons I just told you about in the autopsy is that they all fell to just trend level after we corrected for multiple comparisons with an FDR correction. And just thinking about this, this may reflect selection bias. So autopsies in general are more likely to occur in individuals that have an advanced stage of dementia, kind of regardless of what group they were in 15 years prior. So I think it is striking that we're seeing any differences at all, looking at neuropsych performance, and that could be related even at trend level to autopsy findings, up to a decade later is pretty interesting. And then I'll just mention these other pathologies. This is a simplification here, but, we looked at Lewy body, which was based on, again, a consensus criteria pathological diagnosis. Vascular pathology, which included macroscopic infarcts, microinfarcts, and hemorrhages. And then there actually weren't that many people in the other group. It was about 28 individuals that included hippocampal sclerosis, FTD, multiple sclerosis, a handful of other things. So definitely there were some comorbid pathologies, vascular in particular was common, but there were no group differences in terms of how often these came up. Okay, and this is probably the part people are waiting for, this direct comparison to the consensus diagnosis. Okay, so what's shown here in the rows are the cluster groups, and then the consensus with normal, impaired, not MCI, and then MCI. So the majority of participants who were in the optimal cognitively normal group and the typical cognitively normal group were also classified as normal by the consensus diagnosis. So there was a lot of agreement here. So 95% and 88%. So that's encouraging that there's some agreement there. There was less consistency in the methods for non-amnestic MCI. So 86% of the non-amnestic MCI group had been classified as cognitively normal by consensus diagnosis. And so it's really not surprising if, the focus of the ADRC has been on amnestic MCI, over the years, with less focus on non-amnestic MCI. So, the group had some executive problems and some visual spatial problems, but memory looked fine. So, that made sense why they would get a normal diagnosis. But then, again, going back to having that group as part of the cognitively normal comparison group, could be problematic if they do have some cognitive impairments. Moving on to the cluster-derived amnestic MCI group. So that actually was pretty split between a consensus diagnosis of normal in 57% of the cases, and then MCI in 41% of the cases. So that's kind of interesting because, we're kind of looking at amnestic MCI in both cases, but there was less consistency in that diagnosis. And then for mixed MCI, the majority, 64%, were classified as MCI based on consensus, but there was about a third of them. There were about a third that were classified as normal as well. And then that impaired, not MCI diagnosis didn't correspond to any of the cluster groups. I thought it might, kind of those ones that are impaired, but the explanation's not clear, was pretty spread across the different clusters. So while the primary source of the discrepancy between the groups was definitely that non-amnestic MCI group, this is showing that there were participants that were captured within our memory impaired groups. So the amnestic and the mixed were capturing more participants than the consensus diagnosis. And these findings are consistent with another study that was just published that was led by Lou Graves in Mark Bondi's lab. And she had used the NAC dataset, so the NAC UDS, and she was actually assigning diagnoses at an individual level using the Jack Bondi criteria. And what she found was that approximately 1/3 of the individuals who had been classified as normal on consensus diagnosis did indeed meet neuropsychological criteria for MCI. So there is this group that's showing some cognitive impairments on neuropsych, but these impairments may not be reflected in things like the CDR, called the dementia rating scale, or they may not be reported as problematic by participants and their family. So it's not enough that they're meeting a consensus diagnosis of MCI. So this is a direct comparison then of survival curves. So on the left is the one I showed you before with the 5 cluster groups. And then on the right, you have consensus diagnosis, normals in blue, MCI in green, and then impaired no MCI in red. And so there definitely is this nice separation in consensus diagnosis between cognitively normal and MCI. So there's no question that that's separating people who progress and who don't. But I think one difficulty, though, in making a direct comparison between these two methods is that there's this confound in that the consensus committee is assigning the dementia diagnosis, but they're also assigning the MCI diagnoses. So the consensus committee may be more likely to call someone demented who they'd previously called MCI, as opposed to jumping right to a dementia diagnosis from normal. So it just adds a little bit of a confound there in terms of trying to compare on progression rates. So the way I thought I would do it instead was to kind of look at it in reverse. So look at the outcome, look at who progressed, and then go backwards and see, were they captured as MCI by cluster versus consensus? And so if I do it that way, so of those who progressed, so that was 172, 77 of them had been classified as MCI by the cluster analysis and 55% by consensus. And you can kind of see the breakdown where 19 of those were a non-amnestic MCI, most were amnestic MCI, and then 36 were mixed MCI. So it does suggest that the cluster method may be capturing more individuals who are at risk of progression later on. But then I just did something similar with biomarkers. So looking at individuals who are biomarker positive, looking back at their diagnosis. So of those who were positive, 58% were MCI by the cluster analysis and 42% by consensus. And then for AD pathology, so looking at those that had a high level of AD pathology, 72% of them were MCI by cluster analysis and 55% by consensus diagnosis. So it does suggest that maybe the cluster analysis is capturing more individuals that are at risk. And then that brings us back to the why, so why the discrepancies, why the differences. If consensus diagnosis is underdiagnosing MCI, one potential reason is that when you use clinical judgment, you may be less likely to call someone MCI if they are not reporting problems, their study partner is not reporting problems. There's just been no report of functional or cognitive change. So even if they show some impaired scores on neuropsych testing, clinical judgment suggests that they may get a normal diagnosis, which makes sense. The other thing is that if the consensus diagnosis may weigh a little more heavily these measures of global cognition, like the MMSE or the MoCA, and so you'll have these individuals that are showing more subtle cognitive changes on neuropsych testing that won't be reflected in these more broad measures like the MMSE, and so for that reason, people just may be less likely to get a diagnosis of MCI when they're showing more subtle cognitive changes, but not reporting changes that would change their CDR or, their MMSE. And there is work, again, this very nice study that Lisa Graves just published looking at consensus versus Jack Bondi. And she found that the CDR scores, which is the subjective measure of participants' listening, were weighted more heavily than neuropsych performance in assigning a consensus diagnosis. So for this reason, there may be some potential advantages of actuarial methods or statistical methods to finding MCI subjects or identifying MCI subjects. So the first is that it may identify, more subjects that have these subtle cognitive changes. So not big cognitive changes that would change your MMSE score, but the smaller, more subtle changes. Kelsey Thomas has done a lot of nice work looking at error variables too. So something like intrusions on CBLT might be enough to show these very early cognitive changes in the preclinical AD phase that we might be able to capture those individuals and follow them over time. The use of actuarial or statistical methods is also, it has the potential to be more consistent in the application. So when you're looking at, especially multi-site types of studies where you're trying to have some consistency in how you make a diagnosis, these neuropsychological methods with, clear cut points, X number of scores that need to be impaired is a little more easy to apply in a consistent way. And another advantage is that it goes beyond just our typical subtypes or single domain, multi-domain, amnestic, non-amnestic. So if you think about the 5 groups that I showed you, if we had been just using those conventional labels, then both the amnestic MCI and the mixed MCI would fall into a multi-domain amnestic MCI because they both had memory changes plus something else, either language or others. And so, I think it was helpful to separate those 2 groups out. The mixed MCI, they had this multi-domain cognitive impairment, definitely the highest rate of progression over time. And then in that subset, they had the highest, CSF positivity and a trend towards more severe AD pathology at autopsy, versus that amnestic MCI group that seemed to be potentially earlier in the course, that had, just the memory and language problems and, less severe pathology, less progression over time. So, separating those may be useful. Are there disadvantages to the actuarial approach? There definitely could be. Excuse me. So there are cases where looking at the clinical context could be very important. Thinking about that, impaired not MCI group. So there are these individuals that are showing low scores for one reason or another, but you don't think it's due to AD or something neurodegenerative, right? So it could be low baseline functioning, it could be cultural factors that affected their performance. So there definitely are cases where that clinical history is important. And so I think maybe, in the end, it may be that actuarial approaches and clinical approaches could be complements to one another. That maybe our actuarial approach could make some initial classifications. And then there may be participants where you would kind of tweak their classification based on something about them. English is their second language. You don't want to put them in the MCI group based on low scores, things like that. So I think they can complement each other, really using more of a statistical approach, but using our clinical context to make some tweaks from there. Okay, and just to kind of make some conclusions here from the study. So results show that actuarial methods can produce reliable cognitive phenotypes with differing cognitive profiles and different rates of progression. And data from a subset of individuals showed that they may have unique biological and neuropathological signatures. I mentioned, we didn't really find any differences between our optimal and our typical groups from the things that we looked at, but I do think that this delineation of subgroups of normal may allow for more exploration of those protective factors. And just incorporating these actuarial methods could enhance diagnostic sensitivity. So really helping us to identify more participants with MCI that we can include in our research studies, and that could be enrolled in clinical trials. And definitely further work is needed. As I had mentioned, it was a largely white and well-educated sample, so that limits generalizability. So really looking at this in more diverse samples will be important to see how the clusters play out in those groups. And then it will just be interesting to look at stability of the cluster groups over time. Really seeing, like, does a participant start off maybe in typical cognitively normal, then they go to amnestic MCI, then mixed over time. I think that'll be interesting to see if they are stages of the disease or if they are kind of separable entities. So that's something we could do in the future. So thank you very much to all my collaborators. And I also just want to acknowledge and thank the ADRC and the VA for supporting all this work.
Thanks, Emily.
Thanks, everyone.
Thank you so much.
Wow.
It was a wonderful talk. We really appreciate your participation.
Thank you so much. Thank you for the invitation.